feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity webinar. Thank you for joining us today. Assistant Professor Shih-Yin Tsai is our guest talking about a very important organ, and that is the muscle. We have more than 650 of it. Please use the Q&A function for all your questions. I think the presentation will trigger lots. As always, we are starting with a short presentation, this time by Sanjay Kumar. He is Research Fellow at the NUS Center for Healthy Longevity, and he will talk about an important topic, and that is cellular senescence.
Thank you for the kind introduction. As mentioned, I am Sanjay, a research fellow at National University of Singapore's Center of Healthy Longevity. Today, I will present to you my research plan on topic dissecting cellular pathway of aging and senescence associated amyloidosis. Aging is the process of gradual decline in physiological function marked by progressive molecular changes over time. When we get older, our cells do not function as efficiently. A group of scientists discovered 12 hallmarks of aging, including genomic instability, telomere shortening, mitochondrial dysfunction, and loss of protein homeostasis. My project primarily focuses on the disruption of protein homeostasis during aging and senescence. I will elaborate on this in the following slides. Senescence can be defined as the biological function of aging that gradually deteriorates the functional characteristic of living organism. During senescence, cells stop dividing, and the accumulation of senescent cell during aging contributes to the progression of age-related diseases. Loss of proteostasis is another hallmark of aging. Imagine our body as a factory producing thousands of products under the supervision of quality checkers to ensure order and efficiency. Similarly, protein are a small molecule in our body that must be folded correctly to perform essential function. This protein folding is regulated by a specific machinery called proteostasis. However, with aging and senescence, this protein folding pathway deteriorates, leading to the accumulation of misfolded clumps in various tissues. These clumps known as amyloid, and their formation is known as amyloidosis. These clumps disrupt the normal cell function and increase the risk of age-related diseases. We don't know the composition of these clumps and how they are associated with aging and senescence. Based on this research gap, we framed our objectives. What are the protein aggregates during the aging and senescence conditions? How they affect the longevity? How can we reduce these age-associated amyloidosis? So to address these questions, we will examine our various cell types, including neurons, kidney, cervical cells, and muscles. To validate our finding, we will utilize animal models such as worms, as well as the tissue and body fluid from the young and old mice, including liver, kidney, brain, muscles, blood, and plasma. Additionally, we will employ mouse embryonic fibroblasts, which are the specialized cell with high flexibility and ability to transform into different cell type as we needed. Senescence will be induced using DNA damaging agent. Furthermore, we will identify, extract, and enrich misfolded protein aggregates. Proteomics method will be utilized to explore novel protein associated with aging and senescence in various cell types. Additionally, we will attempt to prevent the formation of these aggregates by reducing the production of these novel protein through gene knockout. Finally, we will investigate various matrices, including lifespan, fitness, protein aggregation, cellular function, and senescence. Here are the primarily findings when we compare young and aged worm. We could see the accumulation of protein clumps in the aged group. Here, day 2 is considered young and day 12 is considered as aged. This observation is also consistent in the aged muscle, mouse lysate, and senescence-induced mice embryonic fibroblasts as shown in dot blot assay. In future, we will identify amyloidosis-related proteins, processes, and pathway in aging and senescence. In long term, it facilitates pharmaceutical targeting of proteostasis-senescence relationship to extend lifespan and prevent age-related diseases. I am grateful to Professor Vincenzo for giving me the opportunity to carry out this research. In addition, I would like to thank everyone in our lab and BK lab member for creating a healthy and supportive environment. Thanks for listening to my presentation. If you want more information, please visit our lab page. Thank you.
Thank you so much for this wonderful presentation, really showcasing that different hallmarks of aging could interact and connect with each other, and also highlighting that the muscle is very important, which is the topic of today's presentation. It's my honor to introduce you to Dr. Shih-Yin Tsai. She is assistant professor at the University of Singapore, and she's also a member of the Healthy Longevity Translational Research Program. She finished her PhD in 2009 at the Ohio State University, and her research since then really focuses on muscle aging, and she investigates the connection between functional decline and structural changes in muscles throughout the aging process. She is the recipient, quite proudly, I think, of the 2024 Yong Lin School of Medicine Research Excellent Award. Shih-Yin, absolutely, it's a pleasure to have you here. We had lots of conversations already. The floor is yours.
Thank you so much. Good evening, everyone. My name is Shih-Yin Tsai from Singapore, and thank you for Andrea for the nice introduction. I had to share my screen. My research of the topic is muscle aging, or you can call it sarcopenia. It's a common gastric syndrome characterized by muscle weakness and atrophy. The fundamental question is, I'm sure everyone cares about what causes muscle aging. But before I diving into the detail, I want to characterize two words. One is driver versus response, because it's very important when we want to study the disease. So the driver is the root cause of a disease, while the response is our body's attempt to compensate for the dysfunction. The understanding with these two will greatly enhance our discussion and help us to remodify the strategy targeting muscle aging. Generally, our muscle strength and performance peak in the early 30s and decline afterwards. However, the muscle mass decline is happen much later and most significant at 65 years of the age and over. Yet, over a long time, muscle size has been the primary target and readout for any intervention of sarcopenia. The concept, or we assume, is that the growth in muscle size will result in a corresponding increase in muscle power. Therefore, the conventional strategy has been focused on the nutrition supplement of amino acid, the resource of protein synthesis, and the signaling pathway that promote protein synthesis and inhibit protein degradation. In skeletal muscle, mTOR is the key cellular signaling sensor activated by nutrition and the growth factor to promote muscle growth. The inhibition of mTOR by stress or prolonged starvation reactivate the catabolic signaling such as protein degradation and induce muscle atrophy. Intuitively, the solution for sarcopenia should be simple: increase muscle hypertrophy by stimulating protein synthesis and prevent its breakdown. Yet, contrary to what we think, clinical activation of mTOR, irrespectively of nutrient availability, is found in the aging skeletal muscle of both mice and humans. Moreover, while developing fine, transgenic activation of mTOR1 in myofiber recapitulate allowed sarcopenia phenotype, including muscle weakness, muscle atrophy, and metabolic dysfunction. In this scenario, could the increased mTOR signaling be promoting the progression of sarcopenia? Then it will be a driver. Or is the upregulation of mTOR signaling response aiming to boost protein synthesis and prevent additional protein breakdown in elderly muscle? To study this, generate transgenic mice. So the transgenic mice we generate is the mTOR is hyperactivated by deleting its upstream repressor TSC in the fully differentiated muscle fiber. If mTOR1 is the driver of muscle aging, then clinical activation of mTOR by this model will accelerate sarcopenia development. But if the mTOR is the responser for muscle aging, continually activating mTOR could potentially increase muscle size and enhance its strength. So that's how our experimental design. And we find that while these mice developing normally, the TSC1 muscle knockout mice, which is we hyperactivate mTOR in the fully differentiated skeletal muscle, they show a many typical feature of sarcopenia as early as 9 months of the age. They have a reduced muscle mass and strength with an increased prevalence of denervation in adulthood. The instability of the structure of a neuromuscular junction where motor neuron communicate with the skeletal muscle has been reported in a nature aging mouse model. And we find that the adult TSC1 muscle knockout mice show many structure instability at the neuromuscular junction site, similar to the nature aging mice, including reduced axon here distribution and a decreased number of post-dynamic myonuclei. And those myonuclei, which are important because they express a specialized gene, which is to support this neuromuscular junction. And also the third typical characteristic is to increase the disconnect acetylcholine receptor cluster. Acetylcholine receptor is the receptor which is on the skeletal muscle, receive the signal of the motor neuron. Molecularly, the sustained mTOR activation indeed increase protein synthesis, but they also suppress the protein degradation, and this causes the imbalance of protein homeostasis, which accumulate damaged protein and organelle because it blocks the catabolic process. For the audience here is probably familiar with the rapamycin treatment for the targeting aging. The treatment of clinical rapamycin treatment to extend healthy aging are mediated in part to inhibition of mTOR-induced phosphorylation of SSK1 and 4-EBP. Activating of mTOR1 signaling cascade, mTOR phosphorylate SSK, leading to its activation and subsequent phosphorylation of multiple proteins involved in the translation machinery. Whereas phosphorylation of 4-EBP by mTOR release its binding from EI4E, freeing EI4E to recruit translation initiation complex and promote peptide translation. So we thought about whether tune down mTOR mediate protein synthesis by either knockout SSK or activate 4-EBP would be affect the mTOR-induced sarcopenia phenotype. We introduced 2 novel mouse lines to study this, offering a unique approach to understand the interplay between mTOR signaling and the sarcopenia. So the first mouse line, which is showing on the right, it has the myofiber specific delete SSK and TSC in the myofiber. And the second line, which is in the middle at the blue here, is a transgenic mice, which is carrying 4-EBP mutation resistant to mTOR inhibition, those yielding an activate form of 4-EBP overexpression in a TSC1 muscle knockout background. So Molecularly, the increase in the global protein synthesis was repressed by either activation of 4-EBP or SSK1 inactivation. But only activation of 4-EBP is able to suppress the cap-dependent translation by sequence the ER4E from assembling activate initiation complex. And most of the genes identified previously translated by the ER4E are responsible for cellular growth and metabolism, which is most of the study was done in the tumor. Even though it seems like counterintuitively, but in our study, suppression of this type of the gene expression by 4-EBP activation protect the mice from the proteostasis stress. So while 4-EBP activation does not relieve mTOR mediate inhibition of UOK, which is the activator of autophagy. Autophagy, this is the essential protein degradation process, actually is able to restore and respond to nutrient sensitivity in this 4-EBP activate skeletal muscle. And with the prolonged fasting, you can see the autophagy was restored. Consequently, the protein homeostasis is improved because we have a reduced damaged protein and also reduce the damaged mitochondria. Therefore, the oxidative stress is also relieved. So besides restoring protein homeostasis, the denervation is also reduced in the 4-EBP activate skeletal muscle. So just a quickly summary of what I have just shown you so far. So uncontrollable mTOR1 hyperactivation actually caused an increase in protein synthesis and a decrease in autophagy degradation. As a result, accumulation of protein aggregate and damaged mitochondria impairs muscle growth and strength. This is phenotype classical sarcopenia signature. However, when we delete SSK1 in this mouse background, despite the global protein synthesis is reduced, but the cap-dependent translation actually is increased, and autophagy is also blocked. So actually, these mice actually show more sarcopenia phenotype than TSC1 muscle knockout mice. Conversely, activation of 4-EBP resets the basal protein synthesis rate by suppressed cap-dependent translation. And unexpectedly, we find that activation of 4-EBP can bypass the mTOR mediate inhibition of the UOK and restore the autophagy flux. Because autophagy flux was fully restored to the control level, consequently, the protein homeostasis is restored and oxidative stress and denervation are relieved in these mice. To further study what is the effect of 4-EBP activation, we find that when we overexpression 4-EBP in the skeletal muscle and we study the young and the old mice, we find that the neuromuscular junction transmission actually is much enhanced in this type of skeletal muscle. And the old 4-EBP mutant mice have also slowed the muscle aging progression. Our genetic study identified that mTOR is the driver of muscle aging rather than the responser. Yet, the mTOR signal is definitely important for muscle growth and development. Rapamycin treatment, acute rapamycin treatment, actually abolished the protein synthesis up on resistant exercise in young men. And genetic inactivation of mTOR1 signaling during the development leads to the muscle atrophy in mice. Even with these benefits for protein quality control, suppressing mTOR actually has a benefit for the protein quality control. However, if we're just using rapamycin without a well-thought output approach, may be too intense for treating muscle aging. I think the fundamental question should be asked underlying muscle aging is why the nutrient sensing kinase complex mTOR1 loses its sensitivity to nutrients. mTOR1 is now acting more like an overworked manager, making all the wrong decisions about his company. In this case, it's our body. Now it's function at the wrong time and not activate enough when it should be. So just recap what is mTOR is doing. mTOR promotes growth and anabolic process, such as protein synthesis, only when it should be. the nutrient and the growth factor is present. However, when there's no nutrient or there is a stress, the mTOR should be suppressed because that helps to reactivate the catabolic process, such as protein degradation, and then thus promote the cell survive. The dynamic mTOR activity governs protein quality control, which is especially crucial for muscle growth and function. So with the enthusiasm of the Rapamycin application for longevity, actually I'm very excited to know that our Center for Healthy Longevity is going to launch the Rapamycin trial in Singapore. However, even though very exciting, but based on our data, actually we suggest that we need to kind of like implant the trial in a much thoughtful way. Maybe it's better to restore the sensitivity of mTOR, which has been shown before in treating immunosenescence in human vaccination response per se. So the Rapamycin treatment should align with the circadian cycle or nutrient regimen, such as we only take the Rapamycin when we are rest of the day. And so therefore we can fully inhibit the mTOR and let the cells start to check their quality control. But when we are activate or exercise, actually we should be off of the mTOR. So then mTOR can be fully function to promote growth. And that's just my simple opinion. And I didn't know I speak so fast. So I didn't put one more important data is, you remember my previous slide, I show you there's a main exercise and I put the 4-APP there. It's because in our mouse model, we find that when we exercise the THC1 muscle knockout mice, we are able to restore the protein homeostasis, muscle strength, and the muscle size. And what we find that the 4-APP actually specific activate in that mouse model. So suggests that the physiology activity, what it does, maybe underneath the molecular mechanism is to activate 4-APP. So using exercise as a dynamic control of mTOR is per se is much physiological way to do it, but for a lot of athletes who couldn't do the exercise, Rapamycin actually is kind of like our hope, which is can help us to restore the muscle health. But I think the way we apply it should be think more carefully. So thank for your attention. I have acknowledged all the person who did the work about this presentation, and I'm happy to answer any question you might have.
Yes, thank you so much, Shih-Yin. Thank you. I really like, let me start with a question about rapamycin and then mTOR inhibition. Very often the trials in humans are done giving rapamycin and other rapalogs once a week and not even daily. What would you suggest to human researchers what to do to preserve human muscle if you're thinking about your ideas about rest, etc.?
Because if I remember correct, the Australia trial for immunosenescence, they give the elderly for daily Rapalog for a short of the time. I remember 2 months or 3 months, such of that, and they have a 2 weeks washout, and then that the elderly to inject the vaccine and to see whether they response. So therefore, I think that washout period is very important. But for the muscle aging, I think maybe we can adapt a similar way like that. So when we're taking the Rapalog, instead of that, the elderly or the people, the young people, take it during the day, which is the mTOR should be fully function. Maybe we should change a little bit about the time we're taking the Rapamycin. And then in the mice model, which has been shown, the Rapamycin on the natural aging mice had to take like 6 months in order to see the effect. But I think at that time for the mouse trial, the Rapalog is with the food. So the mice is keep taking the drug. So maybe that's why it needs so long to see the effect. So if we can align the normal mTOR activity and just trying to make it more sensitive, we probably can see much strong effect. And also, I think the clinical trial, which is at least one of them in the US, is accompanying of rapamycin and exercise. So that might be also another way to try to boost the mTOR sensitivity.
Yeah, let's come back to mTOR in a second, but now look a little bit broader. Also for the audience who might not be molecular biologists. You want to solve the huge problem of sarcopenia. If we are thinking about the prevalence of sarcopenia, which is 30% in older individuals at the age of 80%, it's a huge, important disease leading to fractures and lots of other negative outcomes in older individuals. We in the sarcopenia field often think about 1 or 2 or 3 problems why sarcopenia actually exists. And very often it drills down to inactivity of the muscle. We are not physically active. That's what lots of epidemiologists say. If you're asking biologists, they talk about denervation, so not having the motor control anymore, a decrease in satellite cells, a lowering of the volume of myotubes. So where do you think really lies the problem to solve sarcopenia?
I think skeletal muscle, it doesn't function alone. So actually, I agree, everyone. So, so the motor neuron depletion and also the inflammation and also the blood vessel refraction, all of them contribute to skeletal muscle atrophy and the weakness. And you have to think about, as you just mentioned, is we know our weakness start at, we are not so strong enough. We know it at start the decline after age of 30, but we only kind of like aware this when we are very old. So if we can functional react this earlier as possible, then we can prevent it. Or what I'm trying to say is, I think skeletal muscle is a very resilient tissue. It has to suck all the problem and is only show his weakness when we are very old. So if we can take action earlier, so then we can solve the problem much earlier. I'm not sure that your question properly. I think it's not only one factor, it's everybody contribute. And the skeletal muscle is such a, excellent tissue, which is he wait for the very end and the crush out. So
yeah. So what you are saying is once we reach the stage of sarcopenia, then the organ is failing because we cannot stand out of a chair anymore. There are falls, etc. But of course, we already know epidemiologically that from the age of 30 onwards, our muscle mass and especially the muscle strength is also declining. However, we do not notice that because it's quite easy that we get out of the chair and that we walk the stairs. And once we are losing so much capacity that we are not able to do the daily tasks anymore. So I think, as you stated correctly, there is a question of timing. When should we actually interact? But I would love to hear a little bit more about the biology also of inactivity if you are studying that, because you have the biological fundamental processes which are changing during lifetime, but then you have an additional fundamental, I would say, a social problem, and that is that our lifestyle is changing, that we are so inactive, and if we are not using our muscles, there will be much, much more breakdown than synthesis of the proteins, much likely. What is your thought?
So because I'm studying mTOR and the skeletal muscle, I think if we can improve the protein quality of the skeletal muscle, is able to send the signaling to the motor neuron as what we show here. So our 4-EBP activation is only in the skeletal muscle, but we are able to restore the communication between skeletal muscle and the motor neuron. So from the muscle perspective of the view, I think if we can improve the protein quality control, we could have a huge impact on the motor neuron healthy and also the blood vessel healthy and also for the systematic information. And that again, so we need to functioners earlier. So if we can have the biomarker like what people do for the insulin diabetes, I think it will help people to be aware. Otherwise, we were just used to this sedentary style and then just let the thing get out of control.
Okay, so you think if we are giving rapamycin to lots of individuals, we will restore the human muscle. Is that right?
Yes, I think it's huge.
Okay.
I think
Okay. There is this recent systematic review from Ayla Hodgkins-Courage and summarizing all the randomized controlled trials in humans, looking at individuals who got rapamycin or other rapalogs versus a control. And there was a positive effect in the immune function, and there was a slightly positive effect in the cardiovascular system. However, the muscle was either not studied or was negative, as many other organs too. So if you would design the next randomized controlled trials in humans using rapamycin and rapalogs, who would you actually include? Is that individuals who already have sarcopenia or who might have probable sarcopenia?
Oh, so actually it's turned out the biomarker for the muscle aging. Actually, we don't have very defined biomarker.
Okay.
Muscle biopsy. Also, we rely on the muscle size, which is I talked about at the beginning. The size actually is not fully truly reflect the muscle quality. So then we have to rely on the performance. But then if on the performance assay for those elderly, which is already very poor, the improvement will be very subtle. But then for the person which is maybe in my age, the middle age, our performance assay is not sensitive enough to pick up the performance. So I think this is another challenge here. How to study muscle aging at early on and also at the very late. What is improvement, so-called improvement? So I think have a defined molecular biomarker like what they have for the immune senescence and also for the cardiology would be actually very helpful to help the succeed of the rapalog trial on muscle aging.
Yeah, I fully agree with you. And let's start with more animal studies, and especially with combining different therapeutics. Mm-hmm. And the therapeutic which we very often prescribe in clinical practice is physical exercise and strength training. So do you foresee doing a study where you do physical activity, you are measuring vitamin D levels, because also vitamin D is very important for the cellular function and especially for replication. So where do you think a intervention which is testing many hypotheses together with rapamycin will fit? Think about combining rapamycin rapalogs with physical activity, with other nutrients, proteins, etc.
You mean to add it at a different time, right? Because rapamycin and the protein supplement actually is one is suppressed mTOR, the other one is activate. Yeah. If we can make them kind of like regimen, like you have the protein in early morning and then have a rapamycin at a very late before you go into sleep, try to manipulate its sensitivity. That is, I also think is, if it's doable, it's going to be much more promising than just apply one.
Okay, so you are really thinking about the timing and not doing it in one approach. Okay, let me switch gears a little bit. We have a multiple number of cells in human muscle and in the surrounding tissues. Think about satellite cells, myotubes, but we also have monocyte macrophages around it. So we have the vascular endothelia cells. Which cell is actually benefiting from your approach, really targeting mTOR the most? And where do you think mTOR regulation is disrupted?
In this case, from our animal study is the muscle, because this is the only thing we manipulate. When we activate mTOR in the muscle, we are able to show the sarcopenia. But when we suppress mTOR in the muscle, when the mice developing sarcopenia, we are able to restore it. So it would be muscle per se, but because I don't have any evidence from the other cellular lineages, so it's hard to say. But it also has been shown before, if you can suppress the mTOR activity in the settler cell, which is the muscle stem cell, we are able to restore the muscle regeneration in the older age. So I think the impact of the rapamycin is multiple cell lineage, and that is all benefit to the muscle health.
Yeah, because also if you're looking at muscle, I think we always define it also as an organ with lots of cellular types, like the satellite cells you already mentioned, which is the stem cell producing many more myonuclei, or injecting a little bit of nucleus into the myotubes to function well. Have you done analysis really looking at your hypothesis within different sort of myotubes, depending, for example, how many nuclei a myotube has, and might that matter?
What's the question is whether it's the muscle type specific?
Mm-hmm.
So in the mice, at least, similar to the human. So when we talk about the sarcopenia in a human, people has been find that it's type 2 fiber, which is glycolysis fiber, affect the most. And we are able to recapitulate that in our mouse model. When we hyperactivate mTOR in the skeletal muscle, we find that it's the type 2B fiber is the most glycolytic fiber get atrophy. So this is at least a good thing, is that we're able to recapitulate. And then when we suppress that, what is the increase restore is the type 2B as well. For some reason, the type 1 fiber does not being affect a lot. But this is also true in a human, that when the human getting older, the type 1 fiber seems to protect themselves from the aging process. And there's many hypotheses. One of them is because type 1 fiber is full of mitochondria. So that mitochondria seems like to protect type 1 fiber from the aging process. But then the type 2B fiber, type 2 fiber naturally doesn't have so much mitochondria. So maybe just some of them got affected, is get more amplify the phenotype. It's just one hypothesis. So hope I answer your question. Oh, and in our model, that when we activate 4-ABP in the skeletal muscle, in the normal mice, actually we increase type 1 fiber. So this mice actually more behavior like marathon running mice. But when we breed these mice to the TSC1 muscle knockout mice, which is we call is a mouse model of sarcopenia, we didn't have that switch. But what is the residue again is on the type 2 fiber. So further suggest that exercise maybe it's not do everything, but it can has another effect to help you muscle strength. Both sides.
I see lots of questions also from the audience coming in, but I have two more for you. The first one is, could you think about an approach to titrate rapamycin or rapalogs to the individual species, an individual, for example, to one mice, based on their mTOR activity? Because very often we just say, we have to upregulate, downregulate, etc. What does that mean? Because it's at a continuous scale.
So I think for the mice, we are easy to get the biopsy from the skeletal muscle with the ethic regulate. But for the human, probably is much difficult. So if we can find some blood biomarker, then we can take the blood at multiple times and to find whether the rapamycin, then we can do a titration of rapamycin in the human. I think for the mice, it's doable, but for the human, we definitely need a biomarker, not only the muscle biomarker, but it's the blood-borne biomarker. So then we'll make the trial much more practical. Okay,
good. One last question before heading over to Janjira, who has lots of questions from the audience. Why are you so interested in muscle? Why not in the brain or in endothelial cells or something else?
It's for the personal interest. Because when I look at my family history, I think I'm going to live to 120. And my family actually doesn't have the neuron problem, but I can see if I behave well, I'm going to live that age. So I better do my muscle better to be healthy. Otherwise, I will have a long life but at the poor health. So part of the reason of my family member, that is a lesson I learned. So that's why I always enthusiasm on muscle actually.
So you really think it's the center. I agree with you that the muscle health will also bring lots of different organs into a healthy state. I would say before you have all the results, go to the gym. That also really helps to bring your muscles up and to be in shape. Over to you, Gindria. So she is a PhD student at the Center for Health and Longevity. I see lots of questions.
Thank you so much.
Yep. Thank you, Andrea. So yes, we have a lot of questions from the audience. So the first question would be, does rapamycin's inhibitory influence on mTOR and concomitant increased myostatin activity actually create a self-limiting effect on muscle hypertrophy? If so, how can we mitigate this?
You mean myostatin?
Myostatin activity.
For myostatin activity, actually, I'm at the question at this point. Because even though the myostatin activity has been shown in the muscle dystrophy disease and the model, but in the natural aging process, the myostatin activity actually has not been shown is responsible for muscle aging. So in this case, I'm not sure it will be relevant. Am I answer your question?
Okay.
So in the disease model, myostatin is important. It has been shown before, and when you inhibit, you restore the muscle size, but not the strength. But in the natural aging research, as far as I know, both in the human and the mice, myostatin has never been part of the story.
So we have another question as well. So would rapamycin be available for consumption for longevity? Is it sold in the market already?
So that's why you had to participate with the Angela's lead, the healthy trial, which is, I think it's going to launch soon. I hope I want to be part of it also. So I think keep in tone, keep in touch. The center is going to launch soon, so please welcome to join.
So we have another question. So what is the relationship between skeletal muscle and the hormone insulin? What is the rate of diabetic patients having sarcopenia compared to other people?
Very good question, because skeletal muscle actually mediate 80% of insulin-mediated glucose intake. So if you have a poor muscle, then your glucose cannot be cleaned out from the blood. Vice versa, when you have developing insulin resistance, most likely the insulin resistant, the most affected tissue actually is skeletal muscle. So if you think about your muscle is developing insulin resistance, he's always in starvation. So therefore he couldn't grow. So that's why the type 2 diabetes patient actually also suffer from sarcopenia much early on. It's because your muscle couldn't grow. It's always under the starvation.
Thank you for the response. Here's another question from Stephanie. So what are the types of physical exercises to prevent or reduce the incidence of sarcopenia?
So far, most of the people suggest resistant exercise for the athlete, because resistant exercise can help you to effectively increase the size. But this does not mean endurance exercise is not good. Endurance exercise actually can give you more profound whole body health, not only the skeletal muscle, but also the cardio muscle and also the smooth muscle. So if you can move, please move both. Both of them are important and will benefit your health, muscle health in general.
So we have a lot of more questions to go. So is it correct that the half-life of rapamycin after oral administration is about 30 hours? So in real-world settings, a person trying to optimize muscle growth and longevity, should they actually avoid resistance exercise within those 30 hours when taking rapamycin?
I think this could work out I think we should listen to the profession, which is doing the human study. I'm so sorry. I'm mainly work on the mice. So I think Andrea will have a better answer for that. But I am more like trying to promote that we should take rapamycin, but at the right time to maximize the effect. Yeah.
So maybe I can kick in. I think both is important to study. First of all, what is the effect of rapamycin, Rapalox on human tissues such as the muscle? And then there's the second part, when should we exercise, of what should we eat, for example, to really grow muscle mass and muscle strength at certain times. So luckily, we have lots of information that strength training, resistance exercise training really helps to grow muscle mass and muscle strength in nearly everyone. We don't have any studies where we don't find a satellite cell who says, okay, now I want to replicate because I got the stimuli, I got a little bit of inflammation around, I'm now going to replicate myself and injecting really nice nuclei into the myotube. So it absolutely works. Now the question, while having a very good intervention for sarcopenia together with a protein supplementation, because you need amino acids to build the muscle, when can we actually really give other drugs such as rapamycin, and what is the timing of it? And I must say, we do not know. We do not have any of these human studies. So as you said, for example, doing the resistance exercise training much more before in combination with getting the proteins in the morning, and then before night time, having rapamycin, Rapalox. On the other hand side, there are many studies, because all the individuals fail to eat lots of proteins, to even give proteins overnight, for example, with a tube. And it also builds the muscle and really builds or stimulates protein synthesis. So the question is absolutely valid. I don't think that we've the answer, but I absolutely think that we can come up with a very nice trial in looking really at the sequence, what to do.
Okay, thank you, Professor Maya, and thank you, Dr. Tsai. So we have more questions regarding muscles and proteins. So regarding proteins, in particular in the elderly that have who have higher incidence of sarcopenia or sarcopenic obesity, sometimes with CKD, so chronic kidney disease, especially those with chronic kidney disease with the GFR less than 60 that need to limit the amount of protein consumption. So based on your expertise, would it be possible to just provide the amino acids instead of actual protein? Any potential blend of amino acids that might work to delay the aging muscle?
I think from my opinion, it's just my opinion.
Yep.
I think to give the protein and the amino acid as equal will have a burden for the kidney and also burden for the muscle, which is already have the hyper mTOR activity. And I think that again go back to what Andrea would just say. I think the combination to have the Rapalox to inhibit mTOR and also have a balance for the protein synthesis is important, because actually, if I am not wrong, rapamycin actually has been shown benefit to the CKD. So suggest that actually suppressed mTOR activity is beneficial. So again, it had to go to like a balance. Yeah.
So maybe for the audience, the guidelines are at the moment doing resistance exercise training, at least 6 muscle groups, 3 times a week if somebody wants to counteract sarcopenia, together with protein supplementation. The protein supplementation is 1.2 gram of protein per kilogram body weight. Now studies are also ongoing to see if specific amino acids or specific molecules should be added, for example, leucin or HMB. So that's ongoing. But what the general guideline at the moment is for the sarcopenia field is really resistance exercise training plus the 1.2, sometimes even 1.5 gram of protein per per kilogram of body weight. So if somebody is 70 or 80 kilos, it's at least 100 grams of protein, 120 per day. And then really muscle grows. But this is in older individuals who are sarcopenic.
Thank you, Professor Maya and Dr. Tsai once again. So we have another question. So are there sex differences in the loss of type 1 and type 2 muscle fiber types with age? And are these changes between animal models and humans actually comparable?
Very good question. This is also very challenging thing we are doing now because I had to admit most of the study was done in the male. And even though in our study, we're also including the female there, but the number wasn't significant enough. So what we have learned so far, we talk about sarcopenia effect on type 2 fiber. Most of the study actually come from the male objective and the male mice. So in our previous study, we find that even with that, so I'm talking about the motor neuron. So in the motor neuron, even though both male and female mice show the decline, but the male show much profound phenotype. The female, for some reason, is protect from the motor neuron denervation. So I would think it's a good news, but for the muscle part, it's not so clear at this point. We are still studying. And yes, for sure, there's a sexual dimorphism. I think everybody in the field trying to catch up as well. And my lab is really thinking about that.
Okay, thank you for the very insightful response. So I didn't know that as well. We have another question. So what is the role of anabolic hormones such as testosterone in maintaining muscle mass?
It definitely is promote the anabolic. So this has been shown in the human and also the mice. But as I think probably people heard of the long-term effect is, so when you short time take testosterone, it does increase the muscle size, it does increase for the muscle strength, but for some reason, actually I also not so clear is that the effect seems like blown out after the 6 months. So if you take the testosterone over the 6 months, you just stay there, you didn't get better, and actually you also decline afterwards, but just a little bit slower than the one doesn't. But then when the study passed the 1 year timeline, everything starts to the normal. So whatever you take testosterone, after the 1 year, everything still decline. So somehow there's a insensitivity development in the human muscle. And why so? Actually, at this point, I don't have any information for that. But if you think about the mTOR perspective of the view, I think because there has been shown the mTOR is downstream of testosterone because testosterone induces the protein synthesis, right? It has to store the mTOR. So if you think about in the aging muscle, if the mTOR, the basal activity is already high, you just make it even higher, but you couldn't turn it down. I think maybe because the protein quality control is not in the check. Therefore, after 1 year, you probably will get the thing even worse. But I think this is need more broad study to carefully examine.
So another question. So which part of the body actually loses the most muscle mass and or strength with age? And does this actually differ between the sexes?
In the sexes, actually, I'm not.
Like the different sexes, so like males and females.
So in general, most of the muscle lose faster is on the leg muscle. So it's on the leg. So for the hands, for the arm, it seems like decline much slower. And it's also, I think it's the male decline much faster than the female, but because the male has the bigger muscle at the beginning, the female has a smaller at the beginning. So maybe that's why when people look at the decline rate, we think, oh, the male decline much faster, the female decline much slower. But if you think about the total, I think the effect is the same.
Thank you. So, okay, let me see. There's so many. So, so here's one. So what are the main differences in effects between a rapamycin and a rapalog?
The rapamycin is the first tier of the rapalog. And people find that because it's the first one we find in the Easter Island, right? And then people do the titration, they find in some cell type, the rapamycin can able to block the whole mTOR activity, but some cell type does not. For example, skeletal muscle, for some reason, when you just grow them in the Petri dish, the rapamycin is much effective to block the SSK1, which is I showed at the beginning. So mTOR downstream regulate protein synthesis, right, to two factors. One is SSK, the other one is inhibit 4-EBP. But in the skeletal muscle, rapamycin more prone to block the SSK1, but not the 4-EBP. Because of that, scientists think, oh, it's not effective enough. So they're developing the second generation or third generation of the rapamycin, try to block the mTOR more specifically, effectively. So that's why the Rapalog is coming. It's just the second or third generation of the rapamycin. And depends on the design, you can totally block the mTOR activity, or mTOR actually, this complex is in two complex. mTOR, this kinase is actually involved in two complex. So am I making things more complicated? So you can either block these two complex or just block one complex at a time, or you can block this complex, the downstream target at a different time. So it's just effectively. For the tumor, if you're taking the Rapalog for the tumor, you want to block both complex. But for the aging, you only want to block one complex, is complex 1. Because that is the one mediate the protein homeostasis, but the mTOR complex 2 is a little bit complex because it's also regulate the structure of your cell and also the insulin sensitivity. So for the longevity perspective of view, you don't want to block mTOR 2, you want to block mTOR 1.
Okay, thank you, Dr. Tsai, for the very technical explanation. Yes, thank you, very informative. So here's another question. So what are the differences in the muscle composition, such as fat, protein, and other nutrients between the different muscle fiber types?
Oh, so we keep talking about type 1 and type 2 fiber. So when you do the resistance exercise, is the big fiber is type 2 fiber, is using glycolysis as a main metabolism. So this is type of fiber like to use glucose. But for the type 1 fiber, we keep talking is full of mitochondria because it has a lot of mitochondria. So actually this type of the fiber has a capacity to utilize the lipids. And this type of the fiber is the marathon runner use. As you probably heard about, the marathon runner actually is leaner because they are more easily to burn out the fat. And that is because the mitochondria. Am I answer your question?
Yes. So does that mean the marathon runner with more type 1 muscle fibers have more lipids in them? Composition of the type 1 fiber.
Much easier to burn out the lipids.
Okay. Okay. Thank you. So let's see. So, okay, if there's a difference in size between muscle fiber types, does it necessarily mean that if someone has a greater composition of a particular muscle fiber type, so in this case, type 2, they would have greater muscle mass and would be more protected than an individual with a smaller composition of the type 2 muscle fiber?
I think let's go back to the original question. What is the sarcopenia and what is the effect of sarcopenia? I think long time people start thinking using the size, but actually size is not matter. It's the strength is matter because the size is very superficial. If you are the marathon runner, you are leaner, but it does not mean your muscle is weaker. On the other hand, those marathon runner, the muscle actually is very strong. So is the strength is matter. So it doesn't matter which exercise you do, as long as you are good on that and keep doing that, make your muscle strong is good.
Okay, thank you. So here's another question. So this is quite interesting. So there are actually biohackers who are self-medicating with rapamycin at the moment.
Oh.
So yeah. So apparently someone has brought this up. So what do you think about the safety and efficacy of these protocols?
Oh,
yeah.
That's why I was waiting for Andrea to launch the trial because if I'm going to take, I want to under the professional guidance. As I say, yes, suppress mTOR, extend the longevity, but don't forget the mTOR actually is also very important to promote the cell growth. So how do you get the balance of that need the professional to guide you? You cannot just try randomly on your own. So that's my opinion. And that need a lot of like evidence. So the Healthy Longevity Center is running on the evidence-based trial. So I think it'll be more benefit.
Okay.
Just try on your own.
Thank you so much for this wonderful discussion. If I just may add, yes, we have randomized controlled trials, but we also have observational studies to actually see if rapamycin users, what they feel, what kind of benefits there are. So Matt Kabeline just reported that a couple of months ago in a scientific journal. There are always pros and cons with medication, and side effects are also there, but we should not neglect the effect. Thank you so much, Shih-Yin. Thank you so much, Andrea, for this wonderful, lively discussion. I think we learned more about muscle this time, so that is great. There will be many more shows where we are also talking about sarcopenia, what kind of trials we at the moment have, and how to measure, for example, muscle mass and muscle strength. So watch our program closely. So for you, thank you so much for participating. It is very valuable to hear your feedback. So if you have any feedback for us, or you think, okay, I would like to learn a little bit about certain topics, or I would love to hear a certain speaker, please reach out to us, and using the chat function, the panelist and all attendees options, please choose that one to leave your feedback. I'm leaving you with a wonderful day. Here in Europe, it's very sunny, with a final video really talking about eating habits and how that might influence our thoughts. Take care.
Most of the weight of your dehydrated brain would come from fats, also known as lipids. In the remaining brain matter, you would find proteins and amino acids, traces of micronutrients, and glucose. Of the fats in your brain, the superstars are omegas 3 and 6. These essential fatty acids, which have been linked to preventing degenerative brain conditions, must come from our diets. So eating omega-rich foods like nuts, seeds, and fatty fish is crucial to the creation and maintenance of cell membranes. And while omegas are good fats for your brain, long-term consumption of other fats, like trans and saturated fats, may compromise brain health. While the human brain only makes up about 2% of our body weight, it uses up to 20% of our energy resources. Most of this energy comes from carbohydrates that our body digests into glucose, or blood sugar. The frontal lobes are so sensitive to drops in glucose, in fact, that a change in mental function is one of the primary signals of nutrient deficiency. Carbs come in 3 forms: starch, sugar, and fiber. While on most nutrition labels, they are all lumped into one total carb count, the ratio of the sugar and fiber subgroups to the whole amount affect how the body and brain respond. A high glycemic food, like white bread, causes a rapid release of glucose into the blood, and then comes the dip. Blood sugar shoots down, and with it, our attention span and mood. On the other hand, oats, grains, and legumes have slower glucose release, enabling a steadier level of attentiveness. For sustained brain power, opting for a varied diet of nutrient-rich foods is critical. When it comes to what you bite, chew, and swallow, your choices have a direct and long-lasting effect on the most powerful organ in your body.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.